Hello everybody, my name is Giulia Ribeiro. I am a PhD student in University of São Paulo and my advisor is Professor Daniel Lar. And for this section of today, I built this poster and I would like to walk through it with you. And I will discuss today how I'm using functional phylogenomics to understand metabolic adaptations in testate amoeba. So, here I show you a picture of our cell intermedia. There is an example of our Arcellinida, and we can see in this picture that they have this outer shell structure that make them useful for environmental diagnosis because people can identify these organisms by morphology of the shell, and this make them really useful for bioindicators. And also, they are versatile organisms because they can occupy saline, eutrophic, and contaminated environments. Arcellinida, they are part of the Amoebozoa group. Here I show you a three-domain reconstruction where we have bacteria, archaea, and I expanded the eukaryotic group. Arcellinida is here together with Amoebozoa. And they are close to Holozoa, the group that includes animals and, well, us, and Holomycota, a group that includes fungi. And the interesting thing about Arcellinida is that it's new, a huge amount of transcriptomic data available for the group that is still unexplored. And most of this data don't have annotation yet. And also there are not many functional studies available. So I would like to show you today how I'm using functional phylogenomics and comparative analysis to understand homology of resistance genes and pathways in Arcellinida lineages. I'm currently working with arsenic resistance, and arsenic is a ubiquitous and toxic element in nature. So most of the organisms are vulnerable to arsenic, but arcellinids and other eukaryotes, a few eukaryotes, have highly resistance to arsenic in the environment. And the current knowledge for this kind of resistance is mainly for the bacterial group. So we know that for bacteria, arsenic resistance is related to the presence of genes in a pathway that take these toxic forms of arsenic and convert to less toxic forms, or to forms that can be eliminated or extruded from the cell. Our hypothesis in this work is that Arcellinida and other resistant lineages of eukaryotes have this arsenic resistance pathways present, and probably they have some differential expansion of these mechanisms. We make this analysis firstly using orthologs because we assume that orthologs have a common origin, and this makes more reasonable to think that these orthologs have the same function. So we build this database using orthologs from Arten cell, genomic, and transcriptomic data. For the homology inference, We take sequences of our gene of interest and we discover their orthologs. And with these orthologs, we build an alignment and clean this alignment and make it ready to build a Hammer profile. And we use this Hammer profile to look for the homologs in our database. With these putative homologs, we build a tree. And this first tree probably still contain the outgroup, which is good because we can use this outgroup to remove sequences that do not belong to our ortholog. And also, we can use it to root our tree and to see the direction of our tree, okay? But we removed this outgroup, and with these remaining sequences, we make a functional curation step where we look, if the sequences have the conserved domains expected, or if they have the active site of the gene, of the enzyme. And we also analyze sequences by distance. So two distant sequences or two similar, like identical sequences, we remove it from our database. And with this remaining data, we built our first tree, which we still need to make more filtering steps after. So what we did, we colored the terminals of this tree by domain. So we colored eukaryotes in blue, archaea in gray, and bacteria in yellow. And we analyzed the clusters. And if we find some strange cluster, like one eukaryotic species in the middle of a bacteria cluster, we analyze this sequence separately. So we make GC content and blast searches, and we decided if we keep or not these sequences in our alignments. And after many rounds of this kind of analysis, we build a final tree that we are confident about the relationships and everything. And with these gene reconstructions, we can identify homology. So we can see, if Arcellinida have a homolog from arsenic resistance genes. Or we can compare eukaryotic lineages with different degrees of resistance. For example, Olomicota, Chlorophyta, and Arcellinida are the most resistant lineages to arsenic. So we can compare them to less resistant lineages and see if they have expansions in these metabolisms or if they have some metabolism that the others doesn't have. We can investigate the origin and evolution of these gene families. And we also can reconstruct the ancestral states. So we can see, for example, if the ancestral of eukaryotes have arsenic metabolism genes. Probably. In summary, we can use this methodology for inferring the functioning of Arcellinida arsenic metabolism. And also we can take these sequences and develop markers to understand more deeply the expression and physiology of these genes in Arcellinida. So that's it. I would like to thank the organization of this protest section, my laboratory partners for the help that they gave me, my fellowship agencies, and I would like to thank everybody for the attention. And I hope everybody is well and safe, and I will be happy for answer any questions that you have. Thank you.